Hello everyone, I'm Dr. Dhruv Kumar, Associate Professor at Amity Institute of Molecular Medicine and Stem Cell Research. My lab is mainly working on cancer metabolism, where we apply cellular and molecular biology approach to solve complex cancer biology problem. So in this 8th Embryology Certification Course for ESHRE exam, I will be talking about basic concepts of cell biology. So if you see, cell is a fundamental unit of life. We all know that. Cell is the study of cell structure and function. So if you see, the overall structural and functional analysis of cell is called as a cell biology. Some organisms have only one cell. That kind of organism is called as a unicellular organism. Whereas some organisms are huge and they have a huge number of cells. That is called multicellular organisms. On the whole, cell biology focuses on the structure and function of a cell, from the most general properties shared by all the cells to the unique, highly intricate functions particular to specialized cells. In 1665, two great scientists, Robert Hooke and Leeuwenhoek, both discovered the cell in plant cell wall in the slices of cork. For the first time they identified. Later in 1830s, two scientists, both were colleagues, friends, Sheldon and Swan, they looked those cells under microscope, both plant as well as animal cells. Then they defined cell and they gave the cell theory. So their definition stated that all living creatures, both simple and complex, are made out of one or more cells. And the cell is the structural and functional unit of life. That is called as a cell theory. If you divide the cell, whole cell structure, it has a subcellular components. And those, the cell biology can be studied in 4 or 5 different sub groups. The one is subcellular compartments, where you study a structure of the cell components, cell regulations, where you study cellular functions, how cell behaves, and cell communication and signaling, that tells you how cells talk to each other in a tissue microenvironment. In cellular metabolism, you study the biochemical and energy regulations of the cell. And in genetics, you study how DNA makes RNA and RNA makes protein, which is essential component to regulate the physiology. If you see, the multicellular organisms have a different types of cells. Some of the important cell types are mentioned here, as you can see: stem cells, bone cells, blood cells, muscle cells, liver cells, fat cells, skin cells, nerve cells, endothelial cells, that is called blood vessels, germline cells, means the sex cells, pancreatic cells, cancerous cells. All these cell types are different cell types, but they have a common DNA. What makes them different is their cellular microenvironment. The relative size of the biological molecules, if you see, it's a different, it varies. So cells can vary between 1 micrometer and hundreds of micrometers in diameter. Within a cell, a DNA double helix is approximately 10 nanometer wide, whereas the cellular organelle called a nucleus that encloses this DNA can be approximately 1,000 times bigger. Now coming to the cellular compartments, there are different cellular organelles are there. If you see the section of cell under microscope, you will be able to see all these subcellular organelles, where there is plasma membrane, microfilaments, nucleus, endoplasmic reticulum, mitochondria, Golgi body, and lysosomes. So normally the pH of this cytoplasm here varies from 7.2 to 7.3, whereas this is the only organelles, lysosomes. It has lower pH in our cytoplasm. What happens in a everyday basis in our cell? Some of the DNAs, RNAs, some of the cell organelles, mitochondria, Golgi body, endoplasmic reticulum, some of the proteins, they get degraded or denatured. So autophagy gets activated. And these lysosomes play a very important role during that autophagy mechanism that is called self-eating process. So during that autophagy, this enzyme, lysozyme, helps to degrade those unstructured proteins, aggregated proteins, and damaged cellular organelles. And those degraded materials, again, they get reutilized during the biomolecular synthesis at cellular level. Relative size of different kinds of cells, as you can see here, it varies. Protein size is normally between 5 to 10 nanometer, whereas the mitochondrial size is 1 nanometer, it varies 1 to 10 nanometer. If you talk about human egg, it varies around 3 to 4 nanometer, 100 nanometers. So all these different cells, different cellular organelles, they have a different size. When you see the composition of cells, okay, here as you can see the bacterial cell composition. The 70% of this particular cell is loaded with water only. And only 30% the cellular component is loaded with chemicals. What does it mean? It means that 70% water regulates these 30% chemicals in our body. And in 30% chemicals, you have 15% protein, 4% small molecules, 6% RNA, 2% phospholipid, 1% DNA, and 2% polysaccharides. More or less similar composition we have in eukaryotic cell. Our cells are, in our body system, we have 70% water only. Now, talking about origin of a eukaryotic cell, it has been hypothesized that eukaryotic cells are originated from prokaryotic cells. A prokaryotic host cell incorporates another prokaryotic cell, and each prokaryote has its own set of DNA molecules. The genome of the incorporated cell remains separate from the host cell genome, and the incorporated cell may continue to replicate as it exists within the host cell, as you can see here. And then over the time, during the errors of replication, or perhaps when the incorporated cell lyses and loses its membrane separation from the host, genetic material becomes separated from the incorporated cell and merges with the host cell genome, as you can see here. And this is how slowly this endomembrane gets developed. And this is how you can see a membrane within the cell that creates a separate compartment. This compartment eventually evolves as a nucleus. So this is how you can see the evolution of eukaryotic cell from prokaryotic cell has happened. Cells never live in isolation. They always need cellular environment, microenvironment, where the group of cells, they live together and they survive, they help each other in cellular survival. So the study of cell communication focuses on how a cell gives and receives messages with its environment and with itself. So paracrine and autocrine signaling they do for the cell-to-cell communication. So cells do not live in isolation, that's true. Their survival depends on receiving and processing information from the outside environment. Whether that information pertains to the availability of nutrients, changes in temperature, or variation in light's level. All these factors that control cell-to-cell communication. Cells can also communicate directly with one another and change their own internal working or internal environment in response by way of a variety of chemicals and mechanical signals work. In multicellular organisms, cell signaling allows for a specialization of a group of cells, and multiple cell types can then join together to form tissues such as muscle, blood, and brain. In single-cell organism, signaling allows population of cells to coordinate with one another and work like a team to accomplish the task. Cells have evolved a variety of signaling mechanisms to accomplish the transmission of important biological information. Some examples of this variety are receptors that allows ion currents to flow in a response to photons. So like during cell-to-cell communication, cells, they communicate to each other through either in response to the external factor or in response to the internal factors. So this is how cells, they communicate to each other through biochemical reaction, through the cellular junction, through the signal transmission. So it is very clear here when the cell which is receiving their own factor, the growth factor to activate the receptor, it's called autocrine signaling. The cell target itself. Here you see in signaling cross gap junction, this communication is happening through this protein. A cell target a cell connected by gap junction. This is called gap junction here, and these two cells are communicating to each other. They are transferring growth factor, cytokines, chemokines. In paracrine signaling, a cell targets a nearby cell, as you can see here. These factors are getting secreted by one cell and it's getting received by another cell. In endocrine signaling, the same factor is getting transferred to the other cell through bloodstream. So these are some main the process how cells in a tissue microenvironment, they talk to each other or they transmit information. As you can see here very clearly in autocrine signaling, these growth factors are secreted by the same group of cells and coming back to the same cells. Whereas in paracrine signaling, these factors are getting secreted by these cells, another tissues or another cell types. So in this, here as you can see how the cell in two different cell types in a same tissue microenvironment, they talk to each other. So this is the example of molecular crosstalk between cancer cells and cancer-associated fibroblast, specifically in solid tumor, how they talk to each other in tumor microenvironment. So here you can see this growth factor, hepatocyte growth factor, interacts with c-Met of other cell types, and then it regulates glycolysis and the production of other growth factors. And these particular growth factors again get secreted by this cell, and it activates the signaling pathways here. Then further it induces the production of SGF, and together it regulates the. So this is how actually cells, they talk to each other through growth factors. This kind of regulations are called paracrine signaling. So here this ligand is interacting with the protein and it's activating this ATP, cyclic AMP. Further it activates this phosphorylase particular enzyme, and this acts as a transcriptional regulation inside the nucleus. And then it activates this phosphorylase, this this transcription factor. This factor binds to the DNA to activate the gene regulation. As you can see here, the mRNA is getting transcribed by the DNA. Now coming to the cell metabolism. Cell metabolism is a set of biochemical processes by which energy is either created or used in the cell. The energy resources in eukaryotic cells are regulated by two well-studied organelles, mitochondria or chloroplast. In plant, chloroplast is the energy source that makes food, and in animal, mitochondria provides the energy. Due to the widely held contention that these organelles, so as you can see here in a human body, when you uptake glucose, it gets metabolized, it produces pyruvate. Then further pyruvate gets metabolized into citric acid cycle, and it goes through the oxidative phosphorylation to produce ATP. So in cancer cell specifically, if you see, this metabolism plays very important role. So this is the demonstration. This schematic represents the metabolic regulation in normal cell, where glucose turns into pyruvate. Pyruvate gets utilized by the mitochondria to produce energy. But in cancer cell, what happens? This glucose turns into pyruvate, and because of the impaired mitochondria, this pyruvate turns into lactate, and this pathway. So in normal cells, glucose utilization is less. In cancer cell, glucose utilization is more because of the impaired mitochondria. Here is the difference you can see in normal cells versus cancer cells with respect to the cancer cell metabolism. Cancer cell metabolism is also very helpful to detect cancer cells in our body, where we use phosphorylated 2-deoxyglucose. You inject fluorine-tagged 2-deoxyglucose to the patient, and then this glucose gets trapped into the stage of the fructose 6-phosphate, and this is how PET-CT scan works to detect the cancer cells. So it has a huge application in energy metabolism as well as in diagnostics and therapeutics. So now coming to the cell cycle and cell division. The cell cycle is defined as the period between successive divisions of a cell. The study of the cell cycle focuses on mechanisms that regulate the timing and frequency of DNA duplication and cell division. In 1950, Howard and Stephen Feld demonstrated that DNA replication took place only at a specific phase of the cell cycle, and that this phase was clearly separated from mitosis. From this understanding, other scientists, other people then identified the 4 characteristic phases of the cell cycle that we call mitosis, gap phase, that G1, DNA synthesis, S phase, and gap 2 phase is called G2 phase. The regulation of cell cycle is conserved throughout the eukaryotes, which has since proved to be the case. The mechanism of division in bacteria differs from the eukaryotes, and the control of their cell cycle is also somewhat different, although again it is linked with the DNA replication, because cell divides during cell division, DNA replicates. So this is the reason why we get the same amount of DNA, the same copy of DNA in daughter cells. Although the cell cycle is highly integrated process, the distinct area of interest within the field study have emerged now. For instance, many genes and proteins that influence the passes from one phase of the cell cycle to another, which have been now identified, and specifically in case of cancer, when their expression is altered by mutation or aberrant regulation, they are usually classed as oncogene. Other proteins that regulates cell cycle division that is called checkpoints. Okay, and there are several tumor suppressor genes that regulates the normal cell cycle regulation that controls the checkpoints. Apart from those, with a clearly regulatory role, many proteins have important function in their aspects of the cell cycle. One is replication of DNA and organelles, which is a fascinating process that includes own repair mechanism and self-eating. That I have mentioned earlier is called autophagy process. Now in the cell cycle regulation, in eukaryotes, the cell cycle consists of 4 discrete phases, just now I have mentioned. The S or synthesis phase is when DNA replication occurs, and the M and mitosis phase is when the cell actually divides. And the other 2 phases, G1 and G2, so-called gap phases, are less dramatic but equally important, because during this maturation happens, in G1 the cell conducts a series of checks before entering to the S phase, and later during G2 the cell similarly checks its readiness to proceed to mitosis. So these 2 phases are very important because in G1 and G2 you have a checkpoint to check, okay, whether DNA is okay, it's not mutated, okay, before entering into the S phase or before entering into the mitotic phase. If any mutation is reported, if the cells they find any mutation, then before entering into the S phase or mitosis, they correct the mutation, then they allow DNA to go. Together, the G1S and G2 phases make up the period known as interphase, and cells typically spend far more time in interphase than they do in mitosis. Of the 4 phases, G1 is the most variable in terms of duration, although it is often the longest portion of the cell cycle. As you can see here, the G1 phase is the longest phase. Cell growth happens here in the interphase. Then it goes to the synthesis phase. So there is a checkpoint here. If anything goes wrong during the cell growth, that doesn't allow that cell to go for the DNA synthesis. And then after this S phase, cell go to the G2 phase. Again, the checkpoints are here. They check properly whether DNA is okay or not, then they allow to go for the mitosis. So the time that you see here, normally in G1 phase, it takes 9 to 10 hours, more or less similar time it takes in S phase, 9 to 10 hours. And in G2 phase, it takes 4 to 5 hours. And in mitotic phase, it takes 1 hour. So there's a different time requires to perform the cell cycle. Normal epithelial, eukaryotic cells, they divide in 24 hours. They replicate in 24 hours. So this is the cell cycle regulation that you see the time is divided based on that. So the G1 checkpoints determines whether all conditions are favorable for cell division to proceed. If it is not favorable, then they don't allow to go further. And there are several factors that influence the G1 checkpoints. The external factors, such as growth factors, play a large role in carrying the cell past the G1 checkpoints. In addition to adequate reserves and the cell size, there is a check for genomic DNA damage at the G1 checkpoint. A cell that does not meet all the requirements will not be allowed to progress into S phase. The cell can halt the cycle and attempt to remedy the problematic condition, or the cell can advance into G0 stage, where that we call resting stage. In G2 checkpoints, the bar's entry into the mitotic phase If the certain conditions are not met, as at the G1 checkpoint, the cell size and the protein reserves are assessed. However, the most important role of the G2 checkpoint is to ensure that all the chromosomes have been replicated and that the replicated DNA is not damaged. So this is the role of checkpoints, to check whether the DNA is going to the next phase is okay, it's the right, it's a correct, it's a not mutated DNA. If the checkpoint mechanism detect problems with the DNA, the cell cycle is halted again, and the cell attempts to either complete DNA replication or repair the damage. In cancer cell, what happens? That the halt here, it's a lock. So what you do, you keep that lock open. So DNA, even it is mutated, even it is damaged, it goes through the next phase of the cell cycle. And this is how mutation comes from one cell to to their daughter cells. Now in M checkpoints, the M checkpoint occurs near the end of the metaphase stage of karyokinesis, and the M checkpoint is also known as the spindle checkpoints, okay, where the cell divides, okay. This is how it works. These are the proteins, the cyclin and cyclin-dependent kinase that regulates the different phase of the cell cycle. And this is the first one checkpoint where all the DNA is being checked before sending those DNA for the synthesis. Second checkpoint is here. If it is everything okay, then it will go for the mitosis. So these are the different complex, protein complex that regulates this process. G1 is G2 and M phase of cell cycle. Mitosis and meiosis. If you see in mitosis, diploid cells replicate chromosomes during S phase and aggregate sister chromatids during M phase so that diploid daughter cells are produced. In meiosis, two chromosome segregation phases, meiosis 1 and meiosis 2 happens, which follow a single round of DNA replication during the pre-meiotic S phase. During meiosis 1, homologous chromosomes are segregated to opposite pole. Sister chromatids then segregated to opposite pole during meiosis 2, which results in the formation of non-identical haploid gametes. So this process, meiosis, is very, very important because all the germline mutation that happens during meiosis only. And if the germline line cells that get mutated, they will carry the same mutation in their progeny. Okay. So in most multicellular organisms, meiosis is restricted to the germ cell that are set aside in early development. The germ cells reside in a specialized environment provided by the gonads or sex organs. Okay. Male germ cells or spermatogonia do not enter meiosis until after puberty. The female germ cells or oogonia stop dividing and enter meiosis within the fetal ovary. Those germ cells that enter meiosis become oocytes, the source of future eggs. The length of cell cycle is highly variable, as I have discussed earlier, even within the cells of a single organism. In humans, the frequency of cell turnover ranges from a few hours in early embryonic development to an average 2 to 5 days for epithelial cells. But normally, for this eukaryotic cell, it takes 24 hours to divide. In rapidly dividing human cells with a 24-hour cell cycle, the G1 phase lasts approximately 9 hours and S phase 10 hours. G2 phase for 4 hours and M phase for 1 hour. In early, So the timing of events in the cell cycle is controlled by the mechanism that are both internal as well as external factor. So that we call intrinsic and extrinsic factors. Cell division is hugely regulated by these two factors. So as you can see here, these are the different phases and the process of the cell cycle. Okay, DNA synthesis happens in a S phase of interphase in both mitosis and meiosis. Synapsis of homologous chromosome formation happens in meiosis during prophase 1. That doesn't happen during mitosis, and crossover happens during prophase only in meiosis. Homologous chromosome line up at metaphase during metaphase, okay, in meiosis, and sister chromatid separation happens during metaphase 2 in meiosis and metaphase in mitosis. Number of genetic compositions of daughter cells. These meiotic cells are haploid, but you get 4 cells. In mitotic cells are diploid, you get 2 daughter cells. So this is how this meiosis and mitosis cell cycle is regulated. So in prophase, chromosomes condensation happens, and that is visible, clearly visible under microscope. A spindle fiber emerged from the centrosomes. Nuclear envelope breaks down during this phase, and nucleolus disappears. In pro-metaphase, this chromosome continues to condense, and then kinetochore appears at the centromeres. Mitotic spindle microtubules attached to the kinetochores, and centrosomes move towards the opposite pole. So this process happens during the pro-metaphase. In metaphase, this chromosome gets more condensed and then aligned at the single plane. Whereas in anaphase, again, this separation starts, and these chromatids, okay, sister chromatids, they pull apart and they go to the pole. In telophase, these chromosomes arrive at the opposite pole and being decondensed. Okay, nuclear envelope material surrounds each set of chromosomes. Okay, in cytokinesis, then when this separation happens, this envelope formation happens, then cell divides through the cytokinesis. Okay. So now the question is, how does the cell cycle affect our daily lives? What is the importance of studying this cell cycle regulation? Nowadays, if you see, cancer is the most killing disease in the world. The most of the death is happening now in current scenario is by cancer only. Heart-related death is second. So most of the cancer development, this cancer that developed through, because of the uncontrolled cell division, because of the broken checkpoints, okay, or inappropriate cell division, often stemming from aberration in normal cell cycle regulation. This is how, if you block, if you break the checkpoint of cell cycle regulation, the cells then they divide in an uncontrolled way. So considerable research is directed to identify alteration in cell cycle regulatory proteins that I have mentioned, cyclin and cyclin-dependent kinase, both as targets for therapeutic intervention and as a biomarkers as well. In addition, the field of stem cell biology is closely linked to the cell cycle regulation because these pluripotent cells can divide slowly over a long period of time. Now, another interesting thing in stem cells in our body system, they hide themselves because of the unique cellular environment. And whenever we require those stem cells to divide, those stem cells, they come out from their, the incubation period, and they divide and differentiate into different cell types. So in normal epithelial cells and stem cells, the cell division happens in a different way. Other areas of current research includes investigating cell cycle regulation in growth of organs and in regeneration, where dormant cells can be switched back into the replicative state. Now, in the genomics, how do cells code genetic information into functional proteins? Okay, this behavior, this process is called central dogma of molecular biology. In genomics, our DNA self-replicates, and then this DNA transcribe the mRNA. That process is called transcription. And further, mRNA gets translated into protein that regulates our normal physiology. And this whole process is called as a central dogma of molecular biology. This is how it works. We have a gene, intron, exon. Here is a regulatory points that we call promoter region, where all this transcription factor binds and regulates the expression of gene. And this is your transcripted mRNA. Now, to make the protein, these introns that we call splicing. The splicing happens with the mRNA, and then a small portion of this non-coding RNA that gets removed, that we call long non-coding RNA. And that mRNA gets translated into proteins, and this long chain of proteins then gets post-translation modification to make a functional protein, and this regulates our normal physiology. So it is not so simple actually. If you study in deep, this genome architecture is a very complex structure. It's not simple only DNA and histone proteins. There are several other proteins. There are several enhancer proteins. The other regulatory proteins are there that regulates the expression of that particular gene. This is the enhancer region of a gene where you, as you can see here, this protein enhancer proteins are getting attached at the place where enhancer proteins are there. Transcription factor will bind, enzyme will bind polymerase, and then it will regulate the gene expression. So in DNA replication, as you can see here, topoisomerase, which is an enzyme protein that binds to the double helix here. And then it starts, the unbinding of that DNA. The DNA helicase here breaks the hydrogen bond between the two strands, okay? And this allows the DNA to unwind into two strands of replication forks. And here, as you can see, this protein, the single-stranded binding proteins, keeps that unbinded DNA straight so that replication can start there. So this is how the DNA replication happens. Okay, here you have a DNA polymerase. This is a replication fork. So from 5 to 3 prime, these small strands are getting synthesized. From 5 prime to 3 prime here, the long strands are getting synthesized. And these small strands are getting attached to the DNA ligase. This is a ligation enzyme that ligate the small the DNA. Okay. So this is how it works from 5 prime to 3 prime here, and from 5 prime to 3 prime here, new strands are getting formed. And this is how DNA replication happens. So what we should remember in DNA replication, DNA helicase are responsible for breaking the hydrogen bonds that join the complementary nucleotide bases to each other. Topoisomerases induces the torsional strain caused by unwinding of a double helix. An RNA polymerase enzyme called primase that synthesized primer, and DNA polymerase does most of the elongation work in addition to nucleotide one by one to the 3rd prime and to the new and growing single strand. That during transcription, 3 steps are mainly involved to make mRNA from DNA: that initiation, strand elongation, and termination. So this is the main steps which are involved during the transcription. Initiation, as you can see here, RNA polymerase bind to the DNA. It starts initiation. Then here, this polymerase gets, these nucleotides, monomer, to synthesize mRNA. When this mRNA gets synthesized, the stop codon is here. It terminates the transcription. So these 3 processes are mainly involved. So what we should remember in transcription is that the process of transcription begins when an enzyme called RNA polymerase attaches to the template DNA strand. And 3 different types of RNA polymerase exist in eukaryotic cells, whereas bacteria have only one. In eukaryotes, RNA polymerase 1 transcribes the genes that encodes most of the ribosomal RNA, and RNA polymerase 3 transcribes the genes for one small RNA plus the transfer RNA that plays a key role in translation process, as well as the other small regulatory RNA molecules. Thus, it is RNA polymerase II that transcribe the messenger RNA, which serves as the template for production of protein molecule. Now in translation, again, 3 steps are involved there: initiation, elongation, and termination. Then initiation happens with the help of ribosome. And then elongation, there are several elongation factors that interacts with and termination happen at the end. So these are 64 nucleotides, okay, multiple codons which are responsible for the synthesis, to code different amino acids, okay. As you can see here, AUG code for methionine, that's always the initiation codon, and chain termination codon we have UAA, UAG, and UGA that we call a stop codon. So when this code comes, then translation gets stopped. So this is how it works: translation, initiation, complex. You have a tRNA, and this methionine is attached with that. And here you have this UAC, which will attach to the AUG nucleotide codon here. And then it will, pull this methionine, and this process goes on and on, okay. And this replace elongation factors, when it will bind to this, it will replace to the older tRNA. And this is how, as you can see here, this methionine and glycine, they get attached to each other, and then it will make the long chain of this polypeptide through this process. So older tRNA gets replaced by this newer tRNA through this elongation factor, this protein that helps in the translation. And this is, this is how your DNA gets, mRNA gets, transcribed from DNA, and this mRNA gets, with the help of ribosome, gets translated into proteins. And this long polypeptide, after post-translational modification, they form the functional protein that regulates our normal physiology. So in translation, what we should remember that the translation of mRNA begins with the formation of complex on the mRNA, and the first 3 initiation factor proteins known as IF1, IF2, and IF3 bind to the small subunit of ribosome. This pre-initiation complex and methionine carrying tRNA then bind to the mRNA near the AUG start codon, forming the initiation complex, and the ribosome moves along with mRNA in the 5' to 3' direction, which requires the elongation factor G in a process called translocation. And at the end, there are 3 termination codon, as I have mentioned earlier, that are employed at the end of protein coding sequence in mRNA: UAA, UAG, and UGA. When it comes, then translation gets stopped. So normally we talk about gene. And but the gene is not something that the only it's some part of DNA, but gene is a specific region on our DNA where you have intronic and exonic regions, and some portion there you have a control regions that we call promoter region, where enhancer proteins, transcription factors binds, and it regulates the transcription of exon and intron. And this is the mRNA that when gets transcribed, and after transcription, you splice this non-coding region, and then you get only coding region that codes for the protein. Okay, so normally 20,000 to 25,000 genes we have on our DNA, and 2,000 transcription factors. Okay, 2 mRNA per hour transcription happens in eukaryotic cells. So normally 17 copies per cell mRNA gets transcribed, and it takes normally 7 to 9 hours. The degradation of mRNA, after 7 to 9 hours, it gets degraded. Okay, so during translation protein, 1,000 RNA binding proteins gets involved. As you can see here, 1,000 proteins mRNA per mRNA formation happens per hour. And almost 50,000 of protein copies per cell gets formed, okay, in our single cell. So yeah, So this is all we should know about the central dogma of molecular biology and how our cells, they regulate the gene expression, metabolism, protein expression, okay, and how the cell structure, the cell function, okay, is dependent on the transcriptional regulations of the cells. As you can see here, the cell divides into these two cell types, and this cell further you have this transcription factor. These transcription, different colors, the different transcriptional regulation, that the different transcriptional regulation gives you the different types of the cells, structurally as well as functionally. So with this, I thank you, all of you. So thank you very much.